Aaron. So my name is Austin Keeler. I am a new assistant professor at the University of Delaware with the Department of Biological Sciences. And as Aaron said, my lab will open this fall, so I'm still in that lab setup stage. So today I'm going to focus this short talk on a technique, mass cytometry, that's going to factor heavily in my research program and that I think is broadly applicable to many other labs. A major part of my postdoctoral work was bringing mass cytometry, which has overwhelmingly been in cancer and immune biology, to study the developing and dysfunctional nervous systems. So over the last decade, single-cell RNA-seq studies have really led the way into expanding our knowledge of what types of cells exist in complex tissues, either during development, in response to challenges in adulthood, or in disease and disease models. In developmental neurobiology, the ability to look at multiple gene transcripts in individual cells has been a really powerful tool to allow us to understand how the brain forms. But while gene expression is very essential to who cells are and how they may be responding, it's not the whole story. The main doers really are the protein, and how much protein is present and or active is the cell state, and that does not always line up with the RNA transcript. And disease mechanism, even in cases of altered gene expression, ultimately comes down to protein problems, whether in expression, localization, signaling, or function. As developmental neurobiologists during my postdoc, with interests in signaling mechanisms that drive normal and aberrant neural development, it was a very natural extension for us to seek to adapt this technique, mass cytometry, which is single-cell protein-based, to characterize and explore neurons' glia from a protein perspective. So briefly, what is mass cytometry? It's a flow cytometry variant that increases the number of possible measurement parameters by using fluorophores and fluorescent detection, but rather than isotopically pure rare earth metals and mass spec rather than the fluorophores, thereby avoiding kind of any issue of spectral overlap that causes issues when increasing the number of fluorophores together. Okay, for some reason it's not. There we go. Okay, so unlike RNA-seq, which reads tens of thousands of transcripts, mass cytometry is limited by the available metal isotopes, which right now is 50 that are currently purifiable and commercially available, but we have up to 100 potentially. We conjugate these rare earth metals then to antibodies, and that allows us to detect proteins or protein activity of interest. So for mass cytometry, we use these single cells, whether they're from dissociated tissue or cell culture. They're then fixed, permeabilized, and stained. And then mass cytometry will give us quantitative measurement of protein expression or signaling in a similar way to Western blot, but with single cells, like each single cell is its own Western blot, with over 40 antibodies that we're being able to assess in each of those cells. Once it's been stained to get to that analysis, we get single cells down to single drops of fluid. They're incinerated to release the rare earth metal, and then mass spec detector reads just the amount of those rare earth metals, ignoring the other ions that are present. And then what this does is it gives us discrete sets of scans that are collated into single events, where the intensity of the mass reads indicates marker expression, just as with any kind of antibody chemistry. And we use a metal DNA intercalator, iridium, to positively label cell singlets. As with single-cell RNA-seq, these multivariate data sets require high-dimensional analysis. So this is an example from my postdoctoral research, which studied the developmental landscape of the peripheral somatosensory nervous system. With nearly 3 million cells and over 500,000 neurons across 2 weeks of development. We currently have a parallel study that's being done in the central nervous system that's currently in revision with over 24 million cells, mostly neural progenitors, neurons, and glia. So even though mass cytometry is limited to 40 or more parameters, they're not just random proteins. We pick which proteins that we really want to look at through the antibodies that we pick. So if you have a characterized cell type markers in your system, that's a well-described system, as we do with the primary somatosensory nervous system, then mass cytometry is really ideal for a rapid and cheap profiling of targeted populations across genotypes, ages, and other multivariate conditions. So for example, in this study that I did as a postdoc, I developed this antibody that could identify all the different neuronal cell types and generally the different main non-neurons that exist in the peripheral somatosensory ganglia that were known to be there shortly after birth. And then with such high throughput data, we were able to discover and then consequently validate with traditional tissue staining a novel, rare neuronal set of types that seem to be niche stem cell pools that even persist into adulthood. Also, due to the lengthy developmental time course, we were able to produce a data-driven model of somatosensory development from that single 6-hour experimental run. And that closely parallels this model that was built from decades of work in the field. However, my primary interest in mass cytometry is its ability to assess dozens of signaling proteins simultaneously in individual cells. That's its real strength. That allows a nuanced assessment of signaling consequences of all the cell types in heterogeneous populations, such as primary cell culture. So for example, in another project that I did as a postdoc, we found a very distinct and specific response in only a single population in a primary culture to a stimulus, whereas the other cells did not express a response. So we took extracellular vesicles that we derived from cultured sympathetic neurons, and then we applied them to primary spinal cord cultures that were comprised of neurons, glia, and non-neuronal cells. And then what we can see as a result in these split violin plots, where the intensity of the signal, kind of more intensity the further to the right it goes. Here we have the response to a control in blue, and then the extracellular vesicles in red to these different signaling proteins. And what we found is that only in these committed immature neurons, but not in neural progenitors, not in glia, not leukocytes, the other cells, we saw this robust reaction to these extracellular vesicles. And in this pilot, we only assessed a dozen different signaling antibodies that we had validated at that time across mostly neurotrophic and death signaling pathways. And since then, I've expanded that pool of validated signaling antibodies to over 24, still mostly focused on these neurotrophic and death signaling pathways for now. But again, I find it so remarkable that in this heterogeneous population, this rare population that we wouldn't be able to see in Western blot, might not even know to look for it with staining, we can pull it out in this high dimensional data set and see a very robust and specific reaction in just that population. But these multiplexed signaling experiments have to be done in cell culture because, unfortunately, the length of time it takes to dissociate neural tissues down to single cells is too long to preserve any kind of signaling state for our single cell mass cytometry. So we can do it really rapidly in cell culture, but not from tissue. But there's an adjacent technique called imaging mass cytometry that pairs these metal-labeled antibodies and mass spec with tissue staining. And that bypasses that limitation of trying to preserve signaling states because we can fix that with perfusion. So once the tissue then is stained, it's laser ablated that releases those rare earth metals to be detected by mass spec. And then that allows us to reconstitute the tissue in 40 different parameters. So I've only started to explore what imaging mass cytometry can do as I wrap up my postdoctoral work, but I'm really excited for what that'll also allow my lab to do. For example, here we have a dorsal root ganglia. So this is what houses the primary somatosensory neurons. And in just one tissue slice, we can look at 18 different parameters at once. Obviously, we can go up to 40, but we only had 18 that worked in this first validation round. While mass cytometry has a lot of potential across disciplines, it's very expensive and time-consuming to build the large bank of validated antibodies, which I think is one of the main reasons that many labs are hesitant to jump into it outside of the antibodies that are commercially available for immune and cancer markers. So I'm really excited to leverage the years during my postdoc that I've invested in building validation pipelines and antibody panels to tackle the questions that I have in neurodevelopment, a lot of them that have been intractable for decades, but then also take it to collaborations across and beyond the nervous system. So I really wanted to focus this first talk on this technique that's going to be a large part of what I can bring to the community and what I'm interested in collaborating with. But I want to just give one quick kind of introduction to the kind of work that I'm going to do. So one project in my lab, one of our initial projects, will examine how primary somatosensory neurons gain their distinct modality, or what precise stimulus individual neurons will respond to, and how that's closely tied to the specialized sensory organs, whether it's in the skin or throughout the body, that those particular neurons innervate. So neurons gain their specific responsiveness after they've actually innervated their target. And I'm going to explore the role of cues that are secreted from peripheral tissues that I kind of hypothesize instruct neurons to mature and to decide what type of a neuron to be. So in experiments that would be way too time intensive without the multiplexable nature of mass cytometry, we will culture these primary somatosensory neurons across different developmental time points, especially at key cell fate decision windows. And then we'll expose these cultures to a milieu of different peripheral cues to simultaneously explore both their cell type marker expression and then the concurrent changes in signaling states very broadly. My preliminary results show that these peripheral cues are capable of changing the cell type, the specific type of a neuron, at least by the cell type markers that they express in subsets of these neurons. So then we will next kind of assess the signaling that actually drives those changes. All right, and with that, I'd be happy to take any questions.